Hello friends, I am Dr. Gadekar Sachin Pandit. Here we are talk about the analytical chemistry. Advanced chromatographic methods of analysis. Our paper number is CHA 392. Here we are talk about section first, mass spectrometry, gas chromatography, and section second is liquid chromatography. Today we are talk about mass spectrometry. Here we are talk about the mass spectrometry. It is the instrumental part. Actually, mass spectroscopy analysis the organic molecules. Here introduction first, then principle, second application of mass spectrometry. Mass spectrometry, it is the use for unknown sample containing different types of metal ions are present, they can be analysis. Here samples is used in the vapor form and ionized to give positive ion or radical, and rarely negative ions are also formed. These ions are separated depending on mass to charge ratio and then finally detected. Lastly, recording is done in the graphical form. This is the separate depending on the mass to charge ratio, and lastly recording done in the graphical form of spectrum by using mass to charge ratio and intensity. Here main advantages of mass spectrometry. First, it is highly sensitive technique. Identification of unknown sample. These mass spectrometry it used as identification of unknown sample, identification of suspended compound, structure investigation of compound, molecule, etc. Suppose compound containing different types of fragments, suppose protein, lipid, organic molecules. These compounds structures investigation we are use this mass spectrometry. Molecular weight of compound can be obtained in this mass spectrometry. Mostly we are used for investigation of reaction mechanism, reaction kinetics, decomposition reaction. We are investigation in these mass spectrometry. These are the few advantages of mass spectrometry. Then we are talk about principle of mass spectrometry. Principle, apparatus is evacuated and then sample is introduced in ionization chamber. This is the ionization chamber, it is the part of instrument. First, whole instrument is evacuated and then this sample is introduced in ionization chamber. In ionization chamber, liquid sample introduced in the form of vapor. At that time there use a heater to sample, liquid sample may be vaporized. But some samples are gases. Gases samples are directly used as it is in ionization chamber. At that time, at this ionization chamber, injection systems are provided because of taking sample or introduce the sample. Injection systems are provided there. Ionization chamber. In ionization chamber, ionization of sample takes place. To form a ion, different type fragmentations are generated in ionization chamber. Ionization chamber samples takes place to form a metal ion. In this, suppose ionization chamber containing the different types of ionization source. Molecules may be ionized by using different types of source. First source is electron ionization. First ionization source is electron ionization. Second is chemical ionization. Third is laser desorption. Fourth is force surface ionization. In these ionization, sample ionize by using electron ionization, chemical ionization, laser ionization and surface ionization. Shortly, we are predict EI, electron ionization, chemical ionization, CI, laser deposition, LD, and surface ionization, SI. In our syllabus, there are two types of sources are studied. First is electron ionization and second is chemical ionization. This ionization of molecule takes place due to electron bombardment. Highly energetic electrons are bombarded on the molecule to produce a metal ion. And these metal ions are separated depending on the mass to charge ratio and intensity. This recording done in the graphical form, and then we are predict the graph of mass to charge ratio, and then we are calculate the mass of sample.